Hi, everyone. I'm Jeremy Wilkinson. I'm PacBio's segment lead for microbial genomics, and I'll be the host and moderator of today's webinar. I want to welcome everyone to this webinar with today's topic focused on the recent advances by PacBio in both the microbiome as well as the plant and animal sciences, with the release of our new products, the Kinnex kits, for both full-length 16S and full-length RNA sequencing on our PacBio long-read sequencing platforms. So we have a lot of great material to cover today, and the presentations portion of this webinar is pre-recorded, but will be followed by a live question and answer session with all of the speakers. You are welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee control panel, and we'll get to them either in the chat or at the end in the live Q&A. We've also uploaded a few complementary pieces of literature to your control panel, so please do feel free to download those and take those with you. They have a lot of great material that covers a lot of the stuff we're talking about today. We will be recording this webinar and making it available in the next few days, so please keep an eye out for a follow-up email with a link to that recording. Immediately following this webinar, you'll receive a brief questionnaire, so please do take the time to fill it out, as it is helpful for us to understand your needs and for us to plan for future webinars. You can also stay up to date on future webinars and other events that we're attending by visiting and bookmarking pacb.com/events. So we have a pretty packed agenda today, so it is a 2-hour webinar. I am joined today by 5 great researchers who I'll introduce before each one of their talks. So there are 5 talks in this webinar, followed by a live Q&A at the end, where my colleague Liz Seng, who is the Associate Director of Product Marketing and also the Product Manager for the Kinnex kits, will be joining us for that live Q&A. So with that, let's get started. First up is my colleague Jake Brandenburg, who is the segment lead for Plant and Animal at PacBio, who will introduce the full-length 16S and rRNA methods, and as well as the new Kinnex kits and how they can be applied.
Hello, my name is Jacob Brandenburg, and I'm the Global Marketing Manager for Plant and Animal Sciences here at PacBio. I'm thrilled to be the first speaker in the webinar lineup today, and my topic is going to be an overview of our current Iso-seq and 16F full-length sequencing methodologies, and I'm going to introduce you to our new Kinnex kits, which will be shipping this week. Before we get started, I wanted to give a quick overview of our hi-fi sequencing technology, which is really the technology that allows these applications to come online. So first and foremost, we use a polymerase as a sequencing engine, and we put circularized adapters on the end of your sheared DNA. And what that allows us to do is it allows our polymerase to make multiple passes around a single molecule. And since our error rates are random instead of systematic, we can align the multiple passes that our polymerase makes around the molecule together. And what that leaves us with is very high consensus accuracy, because we can self-correct the individual reads with the consensus of multiple passes. So not only are we left with very high accuracy reads, but those reads also are long reads. So they're tens of kilobases in length, and they can span insert sizes as low as 500 base pairs up to 200,000 base pairs in lengths. You also get single molecule resolution, and this is really important because that allows you to phase a single molecule, and this becomes important when you're looking at assembly and other types of applications. First, I want to start with an overview of our Iso-seq methodology. So when you hear Iso-seq, we're talking about PacBio's RNA sequencing methodology. The reason we call it Iso-seq is because you get a full view of isoform sequencing, hence the name Iso-seq. And if we compare this to what you see on the short read side, with short reads, you're only going to get a partial view of isoforms because those reads are not going to be long enough to span all junctions. So not only are you going to lose a lot to the filtering process, but it can become very challenging when you get to the assembly side. You have to make a lot of assumptions to really parse out that data. With our Iso-seq methodology, we have to see the 3', the 5', and also the poly A tail. So once you do the bioinformatics, you're confident that what you're left with is a full-length isoform, and this is very powerful for annotation and discovery applications. And you don't just have to take my word for it. Over the last year, there has been a ton of publications out there that you can check out that utilize our Iso-seq methodology to generate very high quality genome annotations. And here are a link to a couple of papers that have come out recently. One I want to highlight is a true genome ant notation. And this is a really exciting paper for us because not only did they generate a transcriptome using PacBio Iso-seq methodology, when they did that, they actually found different errors with the existing model. One of them where there were two incorrectly separated gene models. And so with this corrected gene model or with this corrected annotation, now they have that high accuracy, but when they went back to do single cell RNA sequencing on other platforms, they were actually able to increase the percent of reads that map back to this high quality transcriptome. So you can see that not only having a high quality transcriptome is very beneficial to the research in terms of making sure the gene model is as accurate as possible, but it also has the benefit downstream of follow-up differential expression experimentation, having higher mapping back to that high quality annotation. Another great publication on ENCODE 4 came out recently. And the reason that this one is very exciting is because it really shows that it's not just structure variations that allow species to really drive uniqueness from one species to the next, but that isoforms are also huge drivers when it comes to uniqueness across diversity of species. So what this paper highlighted is that not only were they able to detect 200,000 full-length transcripts, 40% of those actually had novel junction chains, but they were also able to find substantial splicing diversity between the autologous genes between the human and mouse models. And this is really what led to that comparison of the uniqueness between the different species because they were looking at autologous gene pairs. And within those gene pairs, more than half of them showed a substantial difference in the mechanisms of the diversity in matching tissue types. So again, just highlighting why we may see significant differences in morphology when it comes to the differences between humans and mice. Now moving on to full-length 16S and why this is important. So as we know, microbiomes really help us answer a lot of important questions about health and disease. Microbiomes are essential for human, plant, and animal species. We know that these organisms perform many services and they also produce a lot of compounds. And we know that these compounds can really have a huge impact when it comes to phenotype, fitness, and ecology of species. And because of these growing matches that we've made, we are now seeing a lot more money and a lot more interest going into funding the study of the microbiome. And the reason for that, and the reason you want to do this with long reads, is because you want to be able to recognize the small differences between species, because that really matters when you're talking about related species. So if you look at the picture here on this slide, the species of penicillin on the left is one that's actually used in blue cheese and is one that it's considered relatively safe and has a lot of uses for compounds throughout different manufacturing facilities. And the compound you see on the right is actually still from the penicillin family, but is a blue mold species and can definitely be related to diseases and can be considered a toxin. So subtle differences in these types of microbes is the difference between something tasting good, but then also something that can be deadly. And so you really need full-length sequencing to get at this. Since this is a fungal species, it's likely in the 18S or ITS space. But the same methodology applies when we look at bacteria getting into that 16S sequencing space. Another publication that highlights the need for full-length amplicon sequencing is a paper out of the Weinstock lab. So this is a paper where they look at different V1 to V9 regions, as well as the full V1 to V9 region. And what they're able to show here is that only when you look at the V1 to V9 region do you see best results, or where you can identify your species back to that species level in 100% of the cases. So you can see the different biases by targeting the different regions here on the left. And again, if you look at the right where that gray arrow is highlighting, you can see that only the V1 to V9 region was able to identify all reads back to that species level identification, or taxa, which we know is ideal for these types of studies. Another publication in the 16S space is comparing PacBio full-length sequencing to different methodologies out there, so such as the V4 and loop-seq. And you can see by the graph here that PacBio outperformed them when it comes to mapping back reads to the phylum, genus, and species level. So 90% of PacBio reads were able to be tracked back to the genus level, and 99.7% of those reads were actually able to be tracked back to the species level. And if, especially at the species resolution, if you compare this back to the other methodologies, you can see that they were only able to detect 31 to 15% of these reads to the species level. So PacBio long read targeting that V1 to V9 region outperforms other methodologies pretty substantially. Now let's get into the meat of the presentation. So introducing the Kinnex workflows and how that's going to be revolutionary for both of these workflows. So when we talk about Kinnex as it relates to these, both full-length RNA isoform sequencing and full-length 16S and other targeted region sequencing uses an amplicon that is a couple thousand kb in length. And as we know, looking at hi-fi reads can be in the 15 to 20 kb range. So if we're only sequencing a single molecule, there's a lot of wasted real estate on that cell. So what we've come up with here is you can take your RNAs and you can take your 16S amplicons and you can put them in solution. And then using a concatenation methodology that's described in the paper you see on the right here, we can actually artificially link together these smaller amplicons into a single hi-fi bar-coded SMRT bell that now contains 8 to 16 different amplicons of interest, all pooled together that can be run across a single SMRT cell on our Sequel to our Revio system. So here's a quick snapshot of what that looks like in the RNA sequencing space. So you can see that this molecule, we're targeting 8-fold concatenation, so 8 different isoforms to build out that Kinnex molecule for the RNA space. We're targeting 40 million reads per Revio cell utilizing the Kinnex kit with our Iso-seq kit. And the Iso-seq kit itself is going to have up to 12 different bar-coded cDNA samples that will be supported at that kit level. And we also have additional bar coding at the Kinnex adapter level if that's necessary for your research. We're also trying to have new standards in terms of depth that is needed to see isoforms. And you can see with the graph down here at the right, at about 10 million reads, we're starting to see saturation where we're getting 80% of known isoforms detected. And we start seeing diminishing returns after you go above 10x coverage, or excuse me, 10 million reads. And so what that means is we know the average transcript length sits around that 1.5 to 3 kb region, depending on the organism that we're targeting. And so you can see that we're starting to hit saturation across the tree of life. If you take a look at the top and bottom graph now, and we see that 80% saturation across the multiple organisms that were a part of this study. So if we condense that down into sequencing recommendation, again, with the goal of being a comprehensive representation of the expressed isoforms in a species. So we're performing genome annotation at this point for plant and animal species. You can also look at multiple tissue types. So we're recommending that you get to 10 million reads per tissue type. So what that means on a single Revio cell now, you can multiplex 4 different tissue types together on a single cell and still hit that 10 million saturation. If you're working with a less complicated species, you can likely get away with less coverage. So you can do 5 million reads for less complicated species, and that will allow you to multiplex 8 different tissue types per Revio cell. And if we take a look at example of what this might cost with the new Kinnex kits, So we have our species of interest, we have our 4 different tissue types, and we're really targeting that 10 million reads per tissue of that species. So we have our 4 different tissues. We each do put those tissue types through a cDNA generation and library prep, and you can see the cost associated with generating those cDNA libraries here. From this point, we can pool these 4 cDNAs together into a single Kinnex prep. So now we're building out those artificial molecules. And then we're going to sequence this Kinnex library on a single Revio cell. And so the all-in cost now comes to $1,950 or $488 per tissue type for an annotation experiment for a single species. And now switching gears a little bit and moving on to 16S Kinnex and what that workflow is going to look like. So again, before you start your project, we support 2 different workflows here. So if you want that high throughput capability, you're looking at the Kinnex workflow. If you want just the standard monomer protocol, we also have that available as well, depending on the number of reads you need to hit per genome. So again, with the standard monomer, you're really running less than 192 samples on our Sequel II system. And with our Revio, you're running around 384 or less samples per SMRT Cell on our Revio. With the Kinnex, this can start increasing much higher folds, and we'll get into that in later detail. But essentially, with Kinnex 16S on the Revio system, you can multiplex up to 1,536 samples per Revio cell. So with the Kinnex kit, again, that's going to lead us to have this scalability when it comes to full-length 16S sequencing. So now utilizing the power of our Revio system and this Kinnex technology, you can do up to 1,536 samples on a single Revio cell, which is really exciting. And depending on the depth that you need to target, you can do that across 1 Revio cell, or you can do this across 4 Revio cells, again, depending on the complexity of the community that you're looking at sequencing. You also have the flexibility of generating these libraries with different adapter sequences, again, for flexibility when it comes to prepping this on the workflow side. And in terms of targeted output, a single Revio cell utilizing Kinnex is going to generate 60 million 16S full-length reads on the Revio system and about 25 million reads on the Sequel IIe system. And since this is still utilizing our full-length cDNA workflows, all of the available tools that you're used to using with our monomer protocol are available on our GitHub page, and you can see the different tools listed down there that you may be familiar working with. Here's a more in-depth look at that cost structure and how it compares across technologies. So you can see that, again, we have different plexity and different throughput levels depending on the size of lab and the size of the experiment that you're running, and the different read depths that you can achieve with those different plex levels across our platforms. So when it comes to cost, if we're comparing our Sequel IIe system to the MAS-Seq, you can see that at all levels of plexity, we are cost equivalent to the MAS-Seq system. And when we compare our Revio to the NextSeq system, you can see that we are cost neutral or cost positive, depending on the plexity. So very competitively priced, very happy with where the Kinnex pricing is rolling up for our Kinnex workflow, and we're really excited to get people to try this out. And so in summary here, if you're wanting to utilize Kinnex with full-length RNA sequencing, you're going to be able to have that comprehensive representation of all expressed isoforms in a given species. So before, we were recommending in the hundreds of thousands read depths on our old monomer Iso-seq protocol, and now you can increase that without increasing the cost up to that 10 million reads, which we're seeing is that saturation point. And that's going to allow you to generate that high quality annotation for any species. And it's also going to start setting us up to look at differential type of experiments now utilizing long read. And so we're very excited for the future of Kinnex as it relates to the RNA sequencing space. If we take a look at our 16S full length offering, you're going to get the best species and strain level taxa that would normally require shotgun metagenomics. So instead of having to do shotgun, you can now utilize full-length 16S amplicons, and pairing that with the Kinnex is just going to make that much more cost-effective, so cost equivalent or cheaper than the short read technologies that you may be familiar with using. So now you can have the extraordinary resolution of full length sequencing with the cost sensitivity that comes along with short read applications. And again, we have different system options. So depending on the scale of the experiments you're trying to utilize, we definitely have a workflow that will match your given research segment. So I want to thank you for your time today. If there are any questions, please don't hesitate to pop them in the chat, and I'm going to be happy to answer them in the Q&A session at the end of the workflow. Thank you so much.
Next up, we have Jeremy Schmutz, who is the co-director of the Hudson Alpha Genome Sequencing Center and is talking on Kinnex full-length RNA for plant and animal research, setting a new standard in high-quality genome annotation.
My name is Jeremy Schmutz. I'm from the Hudson Alpha Institute for Biotechnology. We work on plant genomes and plant gene analysis. We also work as part of the Joint Genome Institute at Lawrence Berkeley Labs in Berkeley, California, where we produce genomic resources for the plant research community for DOE and beyond. I'm going to talk to you today a little bit about the beta that we did for Kinnex. This is a full-length RNA sequencing around plants. I just want to thank PacBio for giving me the opportunity to talk a little bit about our results from that beta and let you know what's going on with this current iteration of the product. So I got pretty excited when I heard that this had been renamed Kinnex because I do happen to have a Phoenix Fury coaster Kinnex that I built with my daughter quite a few years ago. So this weekend, my 12-year-old got it out together, and we thought we'd try to build a DNA helix out of it. And so we did manage to get to the cut site, GAA-TTC, which is a EcoR1 cut site. Which is my favorite cut site. It's the one that we used a lot to build back libraries back in the days of the Human Genome Project. However, Kinnex is actually pretty difficult to work with as a building product. I mean, really, this one says 9+, but I'd probably rate it like 18+ for the amount of effort it takes to build this amazing thing downstream out of Kinnex. So after spending some time working on that, I thought about, well, how can I talk a little bit about Kinnex on Kinnex? So this is my PacBio Kinnex with Kinnex. Explaining a little bit about what this Kinnex technology is. So you start out by generating a bunch of full-length cDNAs and capturing them, and these are my pile of Kinnex that represent those. And then you split them up, in this case, into 8 separate wells, and each one of those wells has a custom set of adapters in it. And those adapters allow the assembly process to occur afterwards. And in 2 of those wells, there's actually the adapters are connected then to the smart belt, the circular adapter on the end that allows PacBio sequencing to occur. You pool all those back together after you've attached those on the ends, and you get something that looks like this coming out the other side, and that's the cDNA that came out the other side. This is this long connected set of cDNAs that you pool together and run on the Revio, and then you break them all back up again and get back out the cDNAs. So it's just a way of kind of making a larger product out of a lot of smaller cDNAs so that you can sample them and then sequence them well on the Revio. So to talk a little bit about annotation, which I want to talk to you about today, I got to talk about where genes come from. So the short answer to where do genes come from is pretty much we make them up. Well, these days we make them up in a more sophisticated way, but in the old days, like when the Human Genome Project happened and we were sequencing our chromosomes, we actually went in and edited by hand the gene structures across all of the human chromosomes. Using this kind of tool, Apollo here, it shows you the ESTs up here and then the gene model down here in purple. Like all good things from the 90s, it's got 90s colors in there. And so you would use it to fix the start sites, the end sites and the stop sites, and then also the introns and extrons along the way. So it was a lot of work to get to that for the human days. Nowadays, we use slightly more sophisticated computer algorithms. This is an example from Soteria viridis, our version 2.1, showing you the genes at the top, the gene structure, with the exons here in beige, and then or pink, depending on how colorblind you are, and then the gray here as the UTRs. And you can see down here, this is supported by these RNA-Seq collections. These are assembled RNA-Seq molecules. And then down here, you see the RNA-Seq quality, the RNA-Seq evidence for each one of those parts of the gene. So this is coming out of our JGI evidence-based plant annotation pipeline developed by Shu and now maintained and improved by Tomasz. And One of the key things here is this PASA assembly step where we're assembling all these RNA-Seq and then trying to pass them through and figure out if they really do match the genome, if they produce a good protein. So when you look at version 4, where we've incorporated full-length cDNAs from Iso-seq from PacBio in there, you can tell quite quickly that, that there's some level of improvement. For example, we've been able to fix this part here, which was confusing, by reading all the way through in full-length transcripts that match the genome exactly. And the reason why it's confusing is because we've actually got an alternate 3' end here, where the assemblies were getting confused, but the full-length cDNAs actually read through it enough to be able to model that structure. So why is Kinnex useful for us? Well, currently we use about 1 to 2 sequel cells mixed with mixed tissues to get to 3 to 6 million cDNAs. For flagships, we add a few more than that. The Kinnex offers up to 10 to 20x the number of full-length cDNAs at a similar cost point, and it could enable us to move annotation to full length instead of RNA sequencing based, the RNA-Seq based. The resolution can also help us to really resolve annotations in our complex polyploids, even haplotype specific annotations, which I'm particularly excited about, and hopefully some new discoveries along the way in expression space, looking at alternate transcripts and some of the larger and more complex genes that are difficult to sample with RNA-Seq alone right now. So the work I'm going to talk about here was done in Jane Grimwood's laboratory. She co-runs the Genome Center at Hudson Alpha with me. The experiments carried out by Melissa Williams in the beta, and then Avinash Sridasayam and my group has done some of the initial analysis. Quickly about the plants, we're looking at Soteria viridis, a green foxtail, which is a small inbred panacoid. It has a 400 megabase genome. It's very compact with 30,000 genes. And you can read more about that in our 2020 paper, Nature Biotechnology. And then our second one is Panicum virgatum switchgrass. That's our biofuel, bioproduct crop, perennial grass, native North America with a 1.2 gigabase genome. We have a haplotype resolved genome for this with about 70,000 protein coding genes per haplotype for switchgrass. And you can read about that in our 2021 paper in Nature. So we ran 4 separate tests for Kinnex. In this, the first one we ran was for Soteria viridis. We did a 4x8, which means it's an 8-plex, 8 concatenated cDNAs with 4 separate tissues that are individually barcoded. We did the same thing for switchgrass, Panicum virgatum, and then we ran a single leaf tissue. That's the one by a concatenation of 8. And then we ran another leaf, the same sample again with 12 concatenation to look at the difference between what you could produce on a single 8-set concatenation versus a 12-set concatenation in the Kinnex protocol. So to start with, you can see the raw yields here on these test ones vary from about 54 or 50 to 100 gigabases. So here you can achieve something like 100 gigabases of data on one of these Kinnex runs here, optimizing the loading and the size of the fragments. After cleanup, we lost about 11% of these 11% of cDNAs that were perhaps malformed, didn't have the right caps on them, or were too low quality once they were extracted out of the concatenated molecules. The library lengths here were between 12 and 14 kb for the 8-plex. They vary because of different tissues going into them and different concentrations of different sizes of expression that would occur within those tissues. And then in the 1x12, that's an 18 kb. So we're really on the edge of kind of what we can capture well with the Revio system. In cDNA world, we captured 26 million to 57 million cDNAs coming out of here. So it is possible to get upwards of perhaps 60 million cDNAs out of a single one of these Kinnex runs. Although it requires some more optimization here on what kind of tissues are going in and the library type that you're using. And the quality scores overall were pretty good. You can see the quality score is down to 1 in 1,000, the Phred score of 30 for the 1x12, and that's due to the number of passes as you push up the molecule size there for PacBio. So looking a little bit deeper into this data here, just showing the difference between full length and full length non-chimeric here, and you can see you can't really see the difference on the left graph at all. On the right graph, that's because the chimera rate is about 1 in 1,000 that comes through the system. And so it's super clean data relative to what we usually produced historically for Iso-seq on the Sequel II. On the genes captured here, this is 5 separate tissues. For Soteria on the top, we capture more than 90% of all the genes that are in the genome. We capture them with full-length cDNAs from the Revio set, from the Kinnex test. And then switchgrass, we capture at least 90% of the genes also. And The reason we don't capture all the genes is interesting. First off, in plants, you don't necessarily get all the tissues that are going to have all the genes that you've predicted in and have evidence. But second, this is really, some of this is a feature kind of the Kinnex system and the full-length cDNA sequencing on PacBio. On the left side here, you can see the curve of the regular annotated genes that are in switchgrass genome here. And then you can see what we captured, which was 54,000 in this one example, and then the missing genes here showing up on the left. And if we kind of zoom in a little bit on those missing genes and kind of look at the curve of captured less than 2 kb, what you can see is that a significant number of these genes that are missing are in the really low end, below 500 bases. And that's because the default Kinnex protocol is really pretty biased against things that are less than 1 kb. So you want to adjust the cleanup step if you want to try to capture a little bit smaller than 1 kb on there. And so that's good for us for annotation because we don't really want a lot of the really small stuff in there. But if you're doing expression or trying to look for a specific kind of length of transcript that's less than 1 kb, you might want to adjust your cleanup procedures for these. So just to mention on the 8-plex versus 12-plex, roughly they look pretty similar on leaf tissue. They look really nice correlation-wise, 99.2% correlation for expression when we're comparing expression of leaf tissue. So there isn't really any specific distribution between those 8 or 12-plex. So I'm going to talk a little bit about preliminary annotation results. This is work done by Tomáš Brune at the JGI, and I just want to say that it's preliminary. I put preliminary on here 4 times, just so you really know that it's really preliminary results. We're just messing around and testing this data. We've only had it for a few weeks. We don't have an updated annotation pipeline that uses this data in the best possible way. So talking about Soteria viridis prelim annotation, the measurement I'm going to talk a little bit about is BUSCO genes. BUSCO is an analysis that was done to identify unique evolutionarily conserved genes across different kingdoms. We use it quite commonly just to express how many of those conserved genes we're actually able to capture with an annotation run. This is a plot showing the BUSCO on here on the left, and then on the horizontal axis, the minimum cluster depth. This is after we've clustered the reads on the genome. The V4 here is blue, and the Revio, that's the Iso-seq, and the Revio is on the top, that's the Kinnex there. And one thing that you can see is the Revio holds up higher cluster depth levels all the way through as you look at higher and higher depth clusters. The Revio really does have a different kind of distribution than what we're getting out of typical Iso-seq in that regard. If you look a little bit at the number of clusters, you can see some of that, what's driving that difference, which is we just have an enormous larger number of clusters coming out of the Iso-seq versus the Revio, and that's just sort of indicative that there's more noise in the original Iso-seq data that we've been using annotation than the Revio Kinnex data that we have. So showing some specific numbers here, the Revio annotation is more complete than even our V4. It has a similar number of cDNAs going into there, although we think the Revio Kinnex are higher quality. We're even achieving 100% of the sort of land plant genes. This would be the widest set, and then 99.7% of the poales, which would be the grasses and sedges set that is really close to what genome here in the switchgrass case, or this is Soteria viridis case that we're looking at. So the higher annotation completeness is driven by more full-length supported genes. Here the Revio is shown in orange versus the blue of the current V4 annotation. You can see we've got quite a few number of genes that here that are covered better in Revio. And looking at that just a little bit differently, so you can see the difference between Iso-seq and the Revio. Now keep in mind there's a lot of different experiments pulled together here to look at this chart. Here showing the current V4 in this blue purple and the Revio in this orange here. So in this middle set here, from about 500 base pairs up to 3,500 base pairs, you can see that we're really accumulating a lot more Revio reads in here, and that's really boosting up the quality of those calls. On the top end, we're slightly reduced on the Revio versus the Iso-seq, and I think some of this is just pressure from the longer molecules, some bias against longer molecules being captured right now in the current 24-hour runs on the Revio. And then on the bottom end, of course, it's greatly reduced in the Revio because we're sort of cleaning out much better on the low end versus what we used to do for Iso-seq here to be able to bias against some of the shorter fragments on there. So some of the example improvements in annotation. On the left here is V4, showing the gene model at the top and then showing some of the evidence below that. You can see in V4, we're basically missing evidence on the side of the molecule here. Over in V5, we're really able to complete the 5' end and get a much better 5' end out because we've got Revio that's lining up there. Here's another example where it's confused in V4 because there's a small gene-like looking object that's sitting in the intron here, and it's breaking up all of our assemblies across there. We're not able to get high quality gene. In the Revio data, we're able to actually produce the whole gene with all of its exons and its 5' and 3' really nice, and recognize that this is actually the anomaly in the middle, and it's not meant to be built in and be part of the genes there. So that's a great capture. And a few more minor improvements that have gone on. For example, a little bit stochastically here in V4, we just don't have enough evidence with this low coverage RNA-Seq to grab this gene here, but we're able to find the gene in the Revio Kinnex and add that back on there. Even in the same, okay, this is showing you how variable the structural annotations really are for gene modeling. Here we're fixing the 3' end here and fixing overlapping 3' ends with the Revio data by getting much cleaner 3' on both of these genes that happen to be overlapping. And we're also fixing the 5' coding sequence on the same gene here by being able to extend a little bit better and higher quality in the 5' end and get more 5' coding sequence. So plans right now for us to improve the gene annotation prediction pipeline is to take better advantage of this high depth of Revio coverage, trying to discard some of the predictions supported by low coverage full-length cDNAs and RNA-Seq assemblies in loci that have higher full-length cDNA coverage. So really trying to get much more accurate consensus out of things where we end up with stacks of these full-length cDNAs and get a highly accurate consensus structures out. Our current implementation uses a global threshold, so it doesn't really utilize the potential of that. So this should lead us to hopefully better filtering of false positive predictions and more reliable alternative isoforms, so we can get all those alternative isoforms predicted and be really happy about the accuracy of those structures. And some of the preliminary tests show some promise. With more stringent full-length cDNA depth, we can remove 3,745 false alternative isoforms out of about 25,000. So we can clean up these alternative, we can improve the base gene models, and then we can clean up the alternatives so we get a really clean versions of these structural annotations. So some of the conclusions about Kinnex, it's a real excellent advance in the number of full-length cDNAs captured with 10 to 20x over the Sequel II Iso-seq with lower cost. So that opens up a lot of opportunities, I think, to use this data. The overall quality of the cDNAs is higher with Kinnex on Revio. We validate it as a drop-in replacement for annotation right now versus the Iso-seq, older Iso-seq versions. Expression studies are likely possible, but you need to be aware of the biases compared to RNA-Seq, and I think more experiments are really needed to determine where you can push the expression calculations here for these. Our experiments weren't really designed to do that. With our improved annotation algorithms, we should be able to provide more accurate structural annotations, and I think with great benefits for our tetraploids or complex genomes that we work with on the plant side. I'm super excited to see what we can learn in polyploids or gene families about variation expression and the biology that really drives it in these organisms by being able to sample and identify the full copies of these genes with the Revio Kinnex that we could never really ever do before with RNA-Seq because they just place all over the place, all over the copy numbers of these. So I did want to mention we work for the Department of Energy Joint Genome Institute, and we do have a public call, which is called the Community Science Program. So please take a look at jgi.doe.gov user programs. We've got lots of opportunities to work with the JGI on that. All of our data that we produce, these genomes and annotations, come through Phytosome, which is a resource that's run and managed by David Goodstein at the JGI. We have hundreds of genomes, now 375 genomes and 163 species to look at, and custom project sites. We also have custom data visualization tools for single-cell transcriptome viewers and a multi-scale Synteny viewer, which has been implemented in the last year to be able to look at these growing numbers of pangenomes that we've been sequencing and analyzing. We've also recently released the JGI Plant Gene Atlas 2.0. This gene atlas covers 2,090 libraries across 18 species of expression data, all analyzed with a single platform. We add biological descriptors to these genes and increase the knowledge of gene function. We also have a great list of genes that need to be prioritized for future experimental investigations, which have lots of interactions but don't have any good functional descriptors. You can access this on Phytosome through Phytosome and also through a dedicated JGI Gene Atlas portal, which has some great tools to be able to interact with that data. So finally, I just wanted to end by saying contact us about JGI user programs or JGI plants. Send me an email or log into the website. If you're interested in PacBio sequencing or applying some of these new technologies to your work, please contact Jane Grimwood at hudsonalpha.org. The work has been made possible from collaborations from Hudson Alpha and Joint Genome Institute and also Arizona Genomics Institute, which provides our biomolecules for PacBio, all funded by the Department of Energy. And if you happen to want a slightly used Phoenix Fury coaster, just let me know. I'll bring it to PAG for you. All right, thank you. Appreciate the time.
Next up, we have Ethan Tai, who is the Microbiome Operations and Services Manager at Zymo Research, who's going to be talking about beyond short read, master microbial analysis with Zymo's full-length 16S microbiome service.
Welcome everyone to this webinar. My name is Ethan Tai. I'm the Microbiome Service Operation Manager. I'm here to talk about the mastering microbial analysis with Zymo Research microbiome services beyond short reads. Let me introduce you to Zymo. We established in 1994 with a humble beginning in a garage. Now we privately own a company with a headquarters in Irvine. We had established our global presence across Germany, Brazil, and China. As an innovation-centric company, we have a strong R&D capability and IB portfolio. Further than that, we also have a strong distribution network of more than 80 partners to establish our international sale. Below is some historic milestones for Zymo. As I mentioned earlier, in 1994, we launched our first product, our Easy Yeast Transformation Kit. In 2012, we actually launched the first NGS-based epigenetic service suite. In 2016, we launched the microbiomic portfolio called Zymo Biomic to help microbiologists around the world with their study. As you can see, at Zymo, we do multiple things, but here today, I want to focus on the microbiomic study and the Zymo Biomic group. With microbiomic, we provide a complete solution from collection to conclusion. Our product portfolio running from microbial standard sample collection specifies for microbiome study, DNA, RNA extraction, library prep, and of course, NGS analysis. We provide all this technology and products so the microbiologists can fulfill their need for their study, but we also combine all of this technology and product to provide a service for customers around the globe. I would like you guys to come with me to a journey for microbiome service as you see how the sample come in and exit our workflow. So when sample come into our lab, the sample will be accessioning for this condition. Sample then store at an appropriate location, either in -80 or at room temperature if they are stored in our proprietary preservative reagent called DNA RNA shield. Sample type is carefully considered at this moment so that appropriate pre-processing procedure can be applied for optimal outcome, whether it is additional removal of PCR inhibitor for soil samples or customized lysis method for tissue samples. Next up, sample will be lysed using mechanical lysis. DNA RNA purification are done on automation platform Kingfisher to allow high throughput processing. For multi-assay of library prep, automation platforms such as Hamilton or TCAN are used for library prep operation. By using automation through robust and calibrated scripting, we can ensure the robustness of the assay, minimize human error batch effect. Also, automation platform allows us to scale up easily and process thousands of samples on a weekly basis. At the end, library would then QC and sequence on either PacBio Sequel2e or any other Illumina sequencing platform. The sequencing data will be analyzed and QC thoroughly with our bioinformatics team to ensure the most accurate result will be delivered to our customer. All this workflow will apply across all the services that we provide to our customer, including our full-length 16S sequencing services. The idea is to simplify the research for customer. They can simply send us sample and we do all the work and provide personalized data report at the end. I want to quickly capture the advantage of using full-length 16S compared to short read. As we are able to sequence and analyze the entire 1.6 kb of the microbial 16S genes, we now have a more complete picture and able to achieve unrivaled species resolution. With 16S, it is more affordable than shotgun method genomic sequencing to achieve the same taxonomy resolution with a faster turnaround time. And amplicon sequencing allows us to analyze a wider range of sample types. Currently, the most popular method for full-length 16S analysis is monomer amplicon sequencing. This process poses some technical challenge. As a service provider, we deal with samples and DNA with various condition and input. Standard workflow is still young and not mature. This shows some difficulty in read normalization, limit the number of samples that we can afford to put in one SMRT Cell. This in turn does not allow us to fully utilize the capacity of the machine, and long read technology, which also drive up the cost for full-length 16S. Enter Kinnex 16S RNA kit that PacBio newly launched this year. With this new technology, and to maximize the advantage of high-fi read length of up to 20 kb. The approach based on MAS-Seq method concatenate multiple full-length 16S amplicon into a 20 kb molecule. These 20 kb molecules are sequenced on PacBio. After read segmentation, we end up with multiple copies of the same 16S amplicon, increasing the throughput of sequencing. With preliminary data using fecal reference standard, the data output shows significant increase from 2.6 million hi-fi reads to a whopping 30 million hi-fi reads. At Zymo, we are very excited to see this improvement. To further testing the robustness of this technology, we send PacBio R&D team an extracted DNA plate with a diverse sample type and condition. This is to mimic the real sample condition my service team handle on a weekly basis. The condition elicits as following. We send them a list of sample with multiple DNA concentration, a variety of host DNA present from human, animal, plant. Sample have a wide range of microbial diversity from low biomass samples such as swab or high biomass samples such as fecal. Also, some sample will have certain level of PCR inhibitor present, and of course, we also include microbial standard because GRACE and required controls. Sample are lysed using beating on a voltage chimney of 40 minutes. DNA purification was performed using Zymo Biomic kit. We choose this combination because it is the most simplified method for unbiased and high molecular weight DNA extraction. Sample then sent to PacBio for library prep and sequencing. the data came back better than we expected. When we comparing Kinnex 16S and monomer 16S data output, hi-fi read output as well as average read per sample increases more than 10 times. When we compare the read output of fecal sample and soil sample, we see a tremendous difference between two approaches. Keep in mind, the library prep process can be further optimized for better normalization. However, as we can see, fecal sample with Kinnex have 10 times more read output, while soil sample have double the amount of read output. With this tremendous, beautiful data, we're looking back the entire workflow holistically to see where we can improve. Just like any other application, metagenomic workflow has a unique set of challenges, especially for long-read applications such as full-length 16S. When collecting sample, we need to be confident that the composition of these sample is not changing between the point of sampling to DNA isolation. We also need to preserve DNA integrity to ensure sufficient quality for sequencing. For DNA extraction, we must ensure uniform lysis of the various micro present to ensure that we obtain unbiased profile. And of course, for long-read sequencing application, DNA length is another key consideration. For library prep, the workflows to have multiple steps, which will increase the hands-on time. The workflow is still needed to be optimized for a streamlined workflow suitable for different sample type. And of course, as important as sample prep and sequencing, downstream application analysis require a robust pipeline with database free of sequencing or taxonomy error for accurate analysis and interpretation. As two pioneer companies in microbiome research, Zymo and PacBio are tackling these challenges together on different fronts. As PacBio keep pushing the boundary of sequencing technology with newly launched Kinnex 16S kit, they have successfully increasing the sequencing throughput at new technology allow to multiplex thousands of samples to be sequenced at the same time while maintaining a high read output with high accuracy. On Zymo's side, we are tackling sample prep challenges. By providing standard control, we aim to fulfill microbiologists' need for their study, such as accessioning workflow bias from DNA extraction to library preparation, or an accurate measuring microbial absolute abundance. Furthermore, we aim to help preserve microbial profile and eliminate cold chain storage and transportation. With DNA RNA shield, we're not only able to achieve those goals, but also preserve DNA fragment size for long-read application. With DNA extraction and purification, we have achieved unbiased lysis with optimal fragment size for hi-fi sequencing. Eliminate the need for additional sonication and cleanup. By using voltectomy with horizontal adapter at max speed at 40 minutes, we're able to achieve complete lysis of all organisms, but the method is gentle enough to retain a DNA length. Testing with fecal sample, we see that we can able to achieve a fragment length upwards of 12 kb. Regarding for library prep, for services, we have optimized and customized the conditions such as primer design and PCR condition so that the workflow is now sample type specific. Coupled with stringent QC protocol, we can ensure optimal result for individual projects. And of course, important as sample prep and sequencing, we continually update our curated database and improve bioinformatic pipeline to get the most accurate analysis. Now you can enjoy all these perks of this optimized state-of-the-art workflow from collection to conclusion at the price of 60-net short read. By simply collecting and send us your sample, we will handle all the work and hand you back a personalized data analysis report of the price of $90 per sample. Thank you very much for your time and happy sequencing.
Next up, we have Chinu Gerard, who is a PhD scholar in early life microbial immunology at the Telethon Kids Institute at Perth Children's Hospital, and whose talk is on the study of dynamics of microbiome diversity in early life.
Hi everyone, I'm Chinu Gerard, first year PhD student from Dr. Archita Misra's Early Life Microbial Immunology Lab. Let me first introduce our lab, which at the nexus of human immunology and microbiology, investigate the impact of early life microbiome in priming or educating the immune system. Especially we focus on the immune memory for long-term health outcomes. So our focus is to understand that the intricacies of this relationship between microbiome and its role in immune memory, particularly with a focus on preterm babies. Today, my talk is about the study of the dynamics of microbiome diversity in early life, where we capture the developmental trajectory from preterm infants in their third trimester to full-term infants in their fourth trimester. Now, before going into its details, let's first understand what is microbiome. Microbiome is defined as the essential collection of all microbes such as bacteria, fungi, viruses, and their genes that naturally live on our bodies and inside us, shaping our life and bodily functions. This in-depth understanding of the microbiome started with the Human Microbiome Project, supported by the National Institute of Health Common Fund from 2007 through 2016. In this Human Microbiome Project, the human microbiome of each and every organ was revealed, and we indeed realized that yes, every organ of the adult body has a unique microbiome. And this was the time when people started thinking microbiome as an additional layer of human body and understood that human body is indeed colonized with diverse microbiome. But how do you think this microbiome is established? To answer this question, we have to go back in early life. And our group has previously analyzed and compared the overall microbial diversity between different fetal organs and their matched negative controls using the 16S rRNA gene sequencing data. First, let's look at the dot plot. The dot plot here shows the distribution of bacterial genera across all sample types. And in the dot plot, bacterial genera that were absent or very low in PBS and high in fetal tissues were denoted as fetal enriched taxa, while bacterial genera high in PBS and low or equal in samples were depicted as PBS enriched taxa. And we observed the enrichment of 7 bacterial genera, specifically enriched in fetal samples. And the next plot focused on the gut, that is the relative abundance plot, showing the significantly enriched taxa in fetal gut as compared to fetal spleen, which was our internal control, and control PBS sample, which was our external control. You can see each sphere, which represents differential taxa, and the size of the sphere represents the percentage relative abundance of the given genera. This comparative analysis of the bacterial enrichment in fetal gut samples in comparison to fetal spleen versus PBS control showed similar pattern with around 22 significantly enriched taxa in fetal gut. These observations suggested a significant enrichment of bacterial signals in fetal organs more notably in fetal gut. And not just our study, several other groups, including Rekhaiti et al., have showed the presence of bacteria-like morphology in pockets of human fetal meconium, and a sparse bacterial signal was detected by 16S rRNA sequencing compared to the environmental controls. Here you can see that 18 taxa were enriched in the fetal meconium, with Micrococcaceae and Lactobacillus as the most abundant ones. Now, although the role of microbiota in immune development in utero is a new and emerging field, decades of research have shown that microbiota is of utmost importance during the first few months of life post-birth, especially in mitigating inflammatory responses in newborn. And this landmark study here showed how important these bacteria are in its functions. Hendrik et al. From Peterborough lab, they showed that a lack of bifidobacteria and their gene depletion required for human milk oligosaccharide utilization, or the HMO utilization from breast milk, is associated with systemic inflammation and immune imbalance early in life. Now, coming back to our study, which focuses on what happens if the baby is born too early. That is, what happens if the baby is born premature. Preterm babies, they get an unequal start to life. And we know that preterm babies, they are more susceptible to infections and they have a weaker immune response. So based on our background information, we propose that as the baby develops, the microbiome diversity increases. But again, what happens if the baby is born preterm is our question. Here, what we are trying is to bridge the gap between preterm babies and full-term babies by testing them with probiotics. For this, we have a very strong literature to support that supplementation with a probiotic mixture accelerates gut microbiome maturation and reduces intestinal inflammation in extremely premature infants. However, all these studies, including our study from our lab, which focused on second trimester, used the conventional 16S sequencing approach, which gives information about the genus level. But it's very important to note that most of the functionality of these bacteria lies in the species level diversity or the species level identity. And what our lab's broader aim is to look for microbe-specific memory, that is the precise immune memory against these early life microbes. Although in this talk, I'll not focus on the memory aspect, but for this microbe-specific memory approach, understanding species level identity is really essential. So to capture this microbiome diversity, we know that there is a gene called 16S rRNA, which is a marker gene for prokaryotes. Its variable region defines the microbiome diversity, and by sequencing it, we get to know the different microbes that are present. So for that, there are two approaches. The conventional approach, which captures only two variables of the full-length 16S rRNA gene and gives the genus level information. But again, in our study, we'll be using a new approach called full-length 16S rRNA sequencing, where you can see it captures all the variables of the full-length 16S rRNA gene and gives not only the genus, but the species level diversity. And remember, most of the functionality of the bacteria lies in the species level identity. So to capture this full microbiome diversity, stool samples of 52 preterm and 14 full-term infants were collected at different time points after their birth, at 1 week, 4 weeks, 4 months, and 1 year. And now after the stool collection, DNA was isolated from the stool. A big thanks to our collaborators, Professor Kai Louis and Dr. Stephen Leach from the University of New South Wales, and their efficient team in collecting the stool samples and isolating the DNA. And we are very much thankful to the Zymo Research Corporation, which helped us in performing the full-length 16S rRNA sequencing to finally obtain the phylogenetic analysis, or the study of species level diversity of different bacteria. That is, the library of whole 16S sequencing was prepared by following the full-length 16S amplification protocol from PacBio. And the pooled library was prepared for PacBio sequencing using the SmartBell Prep Kit. Now, before going into the analysis part, let me first show in specific how probiotics were introduced in the preterm babies, that is in the 52 preterm babies that was considered in our study. So it's a longitudinal cohort study wherein out of 52 preterm babies, 10 preterm babies were given probiotics and the rest were not given probiotics. This was compared with 14 full-term babies. Again, for all the babies, the samples were analyzed at 4 different time points after birth, at 1 week, 4 weeks, 4 months, and 1 year. Now, coming to the results, starting with the taxa composition plot, which illustrates the microbial composition at different taxonomy levels from phylum to species. You can see this interactive figure, which shows the microbial composition at species level, wherein this green color represents the preterm babies that are not given probiotics, and blue represents preterm babies who have been given probiotics, and red represents the full-term babies who were not given probiotics. And from these two figures, which represent preterm and full-term babies at 1 week and 4 weeks, top 5 bacterial species were picked up, and it was observed that the probiotic supplementation in preterm infants aligns their gut microbiome closer to that of the full-term infants. As you can see, the overlap of Bifidobacterium breve, Bifidobacterium longum, and Klebsiella oxytoca as the infant ages. So that's about the taxa composition plots. Next, coming to the alpha diversity analysis, which is a measurement of the microbial diversity of each sample. The plots show the number of observed species in the sample. And the x-axis, again, the green represents preterm babies not given probiotics. Blue represents preterm babies who have been given probiotics. And red represents full-term babies without probiotics. So from these data, we could conclude that as the infant ages, that is from 4 months to 1 year, the probiotic use narrows the alpha diversity gaps between preterm when compared to the full term. While you can see there is a significant difference in the microbial diversity in preterm babies not given probiotics versus preterm babies given probiotics, which suggests that probiotic supplementation increases the microbial diversity and abundance of species. Next, looking at the beta diversity plot. Beta diversity is the measurement of microbial diversity differences between samples. And the figures here are the 3-dimensional principal coordinate analysis plot created using the metrics of paired-wise distance between samples calculated by the Bray-Curtis dissimilarity using the unique amplicon sequence variance. Now, from this plot, we can come to an understanding that the probiotic use in preterm, which is in orange color, leads to a progressive shift in beta diversity, becoming more similar to that of the full term, which is in red color, as the infant ages. This again suggests that the preterm babies given probiotics show a microbial composition more closely resembling that of the full-term babies, indicating a beneficial shift towards healthier gut flora. Now, by this, we conclude that given the preterm babies' heightened vulnerability to infections, understanding that supplementation of probiotics to these preterm babies might unlock new preventive strategies, ensuring an equal start to life. With this in mind, we move forward with our vision to understand and map the early life microbiome, investigate the effect of early life microbial exposure on neonatal immune defense, and understand the early life immunity and disease tolerance against infections and allergies. By this, I would like to thank my supervisor, Dr. Archita Misra, and all my lab members, my collaborators, Professor Kai Louis and Stephen Leach at UNSW, and the PacBio Zymo team for making the study feasible by choosing us as the winner for the Zymo and PacBio Metagenomics webinar giveaway. Thank you.
So this last talk is from my colleague, Jocelyn Braun, who is a senior staff bioinformatics scientist at PacBio, who's going to be talking about enabling high throughput full-length RNA and 16S sequencing with Kinnex kits.
My name is Jocelyn Braun. I'm here to talk about the analysis of Kinnex full-length RNA and 16S rRNA data with SmartLink. So first, I'd like to start with a brief review of our Kinnex library prep workflow, which was introduced in the previous talks. So to start, we need to generate amplicons either from cDNA or 16S. And these amplicons are barcoded. This is denoted by the C1, C2, C3 blocks on the diagram. Next, we use our Kinnex PCR to concatenate these amplicons and create one long molecules. This process introduced segmentation sequences, which are denoted as A, B, C, D, E on the diagram. And finally, once concatenation is complete, we can attach our Kinnex adapters to the end of the concatenated products. And so these are the barcodes plus the smart bell denoted on both ends of the molecule on the diagram. Finally, these libraries can be cleaned up and sequenced. As noted in previous talks, one of the major advantages of Kinnex is to increase throughput. Here we show exactly how much more reads we can get when comparing regular and Kinnex library preps on both Sequel II and Revio, looking across several smart cells. Note that these numbers are for high quality samples, specifically UHRR for the full-length RNA and MSA 1003 for Kinnex 16S rRNA. For the full-length RNA, Kinnex generates about 5 to 6 times more reads than the regular library prep. So that translates to about 20 million S reads on Sequel II or 50 million S reads on Revio. For the 16S rRNA kit, this is a 7 to 8 increase from the regular library prep, which translates in 23 million S reads for the Sequel II and a smart cell and 65 million S reads approximately for the Revio smart cell. So to utilize all this additional throughput, we wanted to enable flexible sample multiplexing. I'm showing here the diagram that was shown earlier with our full concatenated molecule. So we have first barcodes on the adapter level on both ends. So these are denoted in pink. 4 different barcodes are provided, and these are common across all the applications. Additionally, there is application-specific amplicon level barcoding. So these are the ones that happen in your PCR early on. There are 12 barcodes for the Kinnex full-length RNA and 384 barcode pairs for Kinnex 16S rRNA. So these are on both ends. Together, if you combine the 2 levels of barcoding, you can get up to 48 Kinnex full-length RNA samples on your smart cell or 1536 Kinnex 16S rRNA samples. So I have more reads, and we can multiplex the samples. And one might ask, well, how many more samples can I get? How many should I put on a cell? For full-length RNA on Revio, you could put 4 samples and you get more than 10 million reads for each sample. And it's worth noting that this is still much higher depth than you would get putting a single sample with our regular library prep on Revio. If you were willing to give up a bit of that depth of sequencing, you could also opt for 8 samples on a Revio, and that would get you more than 5 million reads per sample. For 16S rRNA, if you're looking at a Sequel II, you can pull either 384 or 1536 samples. 1536 samples give you about 10,000 reads per sample, and the 384 plex would give you much higher depth, about more than 40,000, and you can see the median would be much higher. If you happen to have a Revio, putting 1536 samples on a Revio would get you more than 30,000 reads each. So I can put more samples on my smart cell. I can get more reads. I can still get a lot of deep coverage. How do I analyze all this data? So this is where the good news comes in, that SmartLink 13 comes with new simplified Kinnex analysis workflows. CCS and the adapter level demultiplexing, so these are the little pink barcodes that we talked about before, can happen directly on instruments. So all you need to do is specify which Kinnex adapter barcodes you used on your samples when setting up your sequencing run. Additionally, you can specify an analysis that can be triggered on SmartLink once the data is transferred off instrument, and we call this auto-analysis. So here, I could trigger as auto-analysis for full-length RNA the read segmentation and Iso-seq SmartLink workflow, which encompasses both the read segmentation and the Iso-seq workflow. So at a push button, during your run setup, you can get everything from start to finish. The read segmentation would deconcatenate the reads, and then for Iso-seq workflow, you would get your primary extraction as well as the amplicon level demultiplexing. You would be clustering your isoforms, mapping them to the reference, and finally getting your transcript classification. So at the end of all this, you would get transcripts with the classification as a result. For 16S, the only auto-analysis you can set for now is this read segmentation. So this could get run directly in SmartLink. Then we do provide amplicon barcode demultiplexing as a utility in SmartLink. It would need to be manually launched, but it is possible to do that. And the 16S analysis pipeline would actually happen off SmartLink, and we do provide a workflow on our GitHub. Here, I'd like to show a bit more what the Kinnex full-length RNA workflow looks like in SmartLink. On the left panel, you can see the interface that allows you to set up your analysis. When selecting the read segmentation and Iso-seq workflow, fields will get automatically populated for the Kinnex full-length RNA adapter and for the primer set. So this would be filled up automatically. And then the reference sets, we provide 2 reference sets included. You could have your own custom reference. So human and mouse are the ones we provide. In this case, I'm looking at mouse data, so I'm selecting the mouse reference. Once the analysis is complete, several results panels are available. In the middle section, I'm showing the results you would get from your read segmentation. So at the top are some metrics around the read segmentation, and you can see that we started with about 8.4 million hi-fi reads. These are fully concatenated. Once you deconcatenate them, you would get about 66 million segmented read for this data set. There's additional metrics around how well your concatenation worked and how many of your reads are fully concatenated, as well as some plots. So I'm showing one of the many plots that are available on SmartLink. And here we're seeing the length of the full hi-fi concatenated read and how many segments were observed. So we have, for the most part, the full 8 segments as expected. On the right panel, showing some of the final results that you get from the transcript classifications. The Iso-seq workflow has many plots and many result files which can be downloaded. I am only showing a very small subset here. On the top, you can see the distribution of the isoforms by classification of the structural category. So you can see we have about 24% that are full splice match, some incomplete splice match, and some novel transcripts. At the bottom is also a plot from SmartLink. This is a saturation curve specifically for known genes in this case. And the idea is as I get more reads, how many more known genes do I observe? And you can see that the shape of the curve would sort of tell you how close you are to saturating. And in this case, the curve flattens quite a bit, and you would see that we are indeed reaching quite deep coverage for this sample. So here we are with more reads, more samples, and a solution. So what's next? The first question one might ask is, how does this compare to our non-Kinnex libraries? So I'd like here to compare some data from regular and Kinnex libraries derived from our collaborator mouse samples. Looking first at the transcript length, we can see that the distributions are highly similar, with Kinnex having a much higher number of transcripts. Now I'd like to compare the number of reads that supports known isoforms. So I'm showing here the fractions of reads assigned to observed known isoforms for the regular and Kinnex libraries. The first thing you might note is that there's a very high correlation in isoform counts between the regular and Kinnex libraries. The other thing that you can see is that we have many more known isoforms that observes our Kinnex libraries due to the deeper coverage that comes with the concatenation. Specifically, as shown by known transcripts, you get about 3 times as many known transcripts as the Kinnex library at the same sequencing depth. So we can get more known isoforms. So what about novel isoforms? Here I'm showing the read level and isoform level classifications at different read coverage. So we have 3 different mouse samples, and they're shown here at 3 different coverages. And you can see that at the read level classification remains pretty similar across the different samples and the different read coverage. So that means that as I sequence deeper, I still have about the same fraction of reads that get assigned as full splice match. However, if you look at the fraction of isoforms across those different coverage, you can see that as the coverage goes up, the fraction of known transcripts goes down, and which are blue and orange, and the fraction of novel isoforms, which are shown in red and green, takes a larger portion of our isoforms. This means that as we sequence deeper, we're able to see more of the novel things. So now I'd like to move to 16S. So what SmartLink enables through its utilities is the generation of fully demultiplex full-length 16S reads, which can then be analyzed with the existing 16S pipeline. As mentioned before, the read segmentation can be triggered in SmartLink or via auto-analysis, but then the amplicon barcode demultiplexing needs to be manually launched in SmartLink. And finally, the 16S rRNA analysis is done off SmartLink, and we provide a workflow on our PacBio GitHub where you can use your favorite workflow. So just as with full-length RNA, you would get multiple result panels in SmartLink, and I'd like to show a few of those. So for the read segmentation, once you select read segmentation, you would select your adapter set. For 16S, we use the MAS-12 kit, which does a 12-fold concatenation. I'm showing the same metric panel that we looked at full-length RNA, and you can see that in this case, we started with about 1.5 million hi-fi reads for this particular sample, which translated into 17 million S reads. You can see that the concatenation factor in this case is closer to 12 since we did a 12-fold concatenation. I'm showing some different plots just for some variability. So you can see on the bottom left plot that most of our reads get the full 12-fold concatenation. And you can look at the distribution of the S reads. So these are after the concatenation. And in this case, we have a very tight distribution around 1500, 1550, which is what you'd expect from 16S full-length amplicon. Once you do the amplicon demux, which here I'm showing you would set up through the demultiplex barcode utility, you could select your primer set. In this case, the Kinnex 16S 384 plex primers. You would have to say that we are looking at different barcodes on both ends. These are combinatorial dual end barcodes. You would assign biosamples to your barcodes and hit go. From there, you can get, again, some metrics and some plots. I'm just showing one of the many plots. As metrics, you could see how many barcodes you saw, how many samples were identified, what fraction of the reads got demultiplexed. On the bottom, showing the distribution of the barcode frequencies, and that gives you an idea of how tight your distribution is in number of reads per sample. So as we did with Kinnex full-length RNA, I'd like to compare some of the regular and Kinnex 16S libraries. So when looking at the zymo-fecal reference, which is a more complex sample, you can see that we have a very high correlation of the species representation between the 2 libraries. This is looking at 100,000 reads for each of the libraries. If I downsample to a 384-plex equivalent on Sequel 2, then you can see that with Kinnex, having the additional throughput allows you for the recovery of lower abundance species. And you can see this smear on the left appear for all the reads, the species that are only identified with Kinnex. Additionally, we also wanted to look at our performance against sample with a known species representation. So when looking at MSA 1003, we can see that we have very high correlation, generally speaking, with the provided species and a slightly higher correlation when using Kinnex. Finally, we also wanted to look at some collaborative samples. And here are the results for some samples provided by Zymo. There were 103 samples in this Sequel 2 run, which yielded a high number of reads per sample. So here you can see that we have a median of 90,000 reads per sample, and some samples had very, very high coverage. I also want to show what that increase in throughput can mean. And I'm showing two samples here where we looked at the number of species identified with different number of input reads. The orange sample looks to be higher complexity. And as we increase the number of reads, we keep identifying more new species. And the curve doesn't seem to have flattened quite yet, even when looking at the full sample. However, when looking at the blue sample, which seems to be lower complexity, you can see that that curve flattens quite a bit more, and having additional input does not necessarily translate in a lot more identified species. So here I'd like to go over some of the results presented in this talk. So first of all, Kinnex allows for a drastic increase in the number of reads in your SMRT cell. This is a 5 to 6 increase in number of reads for full-length RNA and a 7 to 8x increase in the number of reads for your full-length 16S RNA. Analysis of this data is simplified by some of the new analysis workflows provided in SMRTlink. This is a push-button solution for Kinnex full-length RNA, where you can go from your sequencing to classified transcript directly from your run setup. For 16S, we are able to generate demultiplex full-length 16S reads from in SMRTlink with a few manual steps. Kinnex also generates comparable data to non-Kinnex libraries. The correlation both for isoform counts or for species representation is very high when you compare regular and Kinnex libraries. Finally, the additional throughput enables higher sample multiplexing. So you could do again, four full-length RNA samples on Revio or 1536 full-length 16S samples on a Sequel 2. Or you can also get a more in-depth view of your samples. So additional throughput allows you to observe more known isoforms or more novel isoforms for full-length RNA. Or for 16S, you can see additional species that are of lower representation in your sample. So with that, I'd like to thank you for your time, for attending and listening to this talk and many of the other talks before. Thank you for the collaborators for providing us with interesting samples. And finally, thank you to the many individuals at PacBio who worked relentlessly to create this product.
Awesome. Thanks, Jocelyn, for that great talk. So now I'd like to invite the rest of the speakers onto camera for us to move into the live Q&A session. We'll also have our Kinnex product manager, Liz Sang, in our live Q&A as well. A lot of you have submitted questions already, so thank you for that. If you do still have some questions, please submit them to the questions area and we will try to address those. Also, I guess you can't hear me now, but if you're having audio issues, you'll need to switch audio inputs to hear the live portion of this versus the recorded. So the first question I have here, I think this would probably be best for Jocelyn or Liz to answer. Is there a way to use the Kinnex Iso-seq data for differential gene expression yet? Would the current kit and protocol support it by including replicates? Is the cDNA amplification step introducing too much bias? And is there a comparison between Kinnex bulk Iso-seq and I can't read the rest, RNA-Seq?
Yeah, let me answer this question. So we have demonstrated with Jocelyn, and this was before Jocelyn's talk, what we really wanted to focus on was that the concatenation with Kinnex does not bias against the original Iso-seq. There's always going to be bias with cDNA generation, right? And as Jeremy showed, if you change how the cDNAs generate, change the bead concentration, you will change the length distribution of the cDNA that's captured. But what we wanted to show was that we're not going to have Iso-seq customers lose what they already get from the Iso-seq data. And I think if you look at differential expression analysis, it's really looking at the ratio of isoform differences from one condition to another. And I actually really like the words now. It's like, as long as the biases are the same, you can detect the signal, right? It's as long as there's a consistent bias. And this actually came from another webinar in a different application where the customer actually said, I'd rather have the same consistent bias than to be unbiased, but in a, well, not unbiased is just, I think, a very difficult challenge, but he'd rather have consistent bias where then you can actually see the racial differences of isoforms. Jeremy, I think the other question was if we have compared this to Illumina. So we have with our collaborator data. I would see if I can, so we're going to have another webinar next year on comparing Illumina versus Kinnex data with our collaborators. We're also hoping to be able to release that matching short read data soon. So just to say that we showed that at ASSG with our collaborators that yes, we have concordance with short read data at the gene level. At the isoform level, not as much, but that's to be expected because we know short reads are not very great at capturing isoform abundances.
Awesome, thanks. Next question, this one I think is for Jeremy. How do you define the chimeric FLNC reads? Is this by gene annotation?
Yeah, it's by when you're putting them back against a high quality genome, then you see the reads are split between multiple gene models, and so that's how. And then the initial Iso-seq versions of that, you have a lot of kind of models that come through that are because. A lot of models that are chimeric that would come through. You'd see reads that are chimeric. The Kinnex seems to have cleaned up quite a few of those sort of historical Iso-seq chimers that come through.
Great. Second question, I think for you or Liz, would you recommend starting from poly-A RNA or total RNA?
I think Liz could probably answer that better than I can. The protocol is total RNA, so that's what you start with.
Yeah, the protocol starts with total RNA. There's no reason to do poly-A selection. It already does it.
Okay. Also, what RNA control materials are used to ensure reliability?
That's an interesting question. So we don't actually typically spike in any RNA controls. We have done them, like we have done it with our collaborators. We've also looked at reproducibility. So not for the early access data, but as part of our data release with our collaborator, we actually generated replicates for multiple collaborators, and we don't see across different species any reproducibility issues.
I'll just keep it on the RNA train for now, and then I'll switch gears to 16S. So this is for, I think, for Jocelyn, or it could be for Liz. For read segmentation of the Iso-seq data, can we add reference sets?
Reference genomes, you mean? I'm trying to understand the question.
I'm assuming so, but I don't know what that would be like. So for read segmentation, it would just be whatever the data coming off the machine is in this case then.
Yeah, maybe there's some misunderstanding of what resegmentation does. It's deconcatenating the Kinnex concatenation, which is predetermined by the kit.
I think the question might relate to the fact that we have segmentation and Iso-seq as one workflow. The reference set that's called on the screenshot is the reference genome. It is possible to add custom reference genome, and there's instructions on how to do so. And we did add a plant genome in our case as a proof of concept.
Cool. This one's also for Jocelyn or Liz. Can I install and use SmartLink software without having a PacBio machine for analyzing publicly available PacBio data?
You want to take that, Jocelyn?
We have a lot of the tools that are available on Conda. So if you wanted to run the different parts of the workflows, you can download and run from command line. That would probably be the better way to try to do that if you didn't want to have SmartLink.
And I'm just going to put in a double down on that. Definitely use the Conda tools if you're going to do analysis rather than trying to load SmartLink and then put in some other data from somebody else into SmartLink and do that. It's just really complicated to do it that way.
Yeah, well, Bioconda is a la carte. Let's put it this way. You can just download what you need. SmartLink, you're downloading all the software with the GUI and the other things that encompasses our applications. It's definitely a monster to install.
Sweet. Easy question here. Does the Kinnex kit start from DNA or RNA?
RNA. So even with the Kinnex full-length RNA kit, you first use the Iso-seq Express 2.0 kit that is also now available to generate cDNA that's then compatible with concatenation. So all Kinnex is based on DNA.
So we had a couple of questions related to this, related to service providers. So it's a great time to talk about it. So, one question was from Germany, the other was from UK. Yes, we have lots of service providers all around the globe. So if you do have a specific region that you're in, that you're interested in getting a local service provider, you can always just reach out to us. Like we have a fill form on pacb.com. You just fill that out and someone will get in touch with you very soon. So that's probably the easiest way to do so. We also have a couple of folks on here that do offer services. So Jeremy from Hudson Alpha, as well as Ethan from Simo Research. So you can reach out to them as well. Going to switch gears to 16S now. I did answer this in the chat, but just kind of voice over it out loud in case folks did not see that. So the question was, how does the cost per sample of full-length 16S compare to shotgun metagenomics? And can you ballpark the cost per sample for full-length 16S if multiplexing in roughly 384 samples? So full-length 16S is actually much cheaper than shotgun metagenomics. And now, like what Jake showed you in his first presentation, was that with Kinnex now with 16S, it's actually equivalent in cost to short read partial 16S. And also what Ethan showed, they're also offering their full-length 16S service for the same price as their partial 16S service with short reads. For 384 samples, it depends on the platform that you're using for sequencing, but at cost, the cost of reagents only, it's roughly around $6 per sample. So very, very, very affordable and cheap. That's USD. The next question also I answered in chat, but am I correct that Kinnex aims to concatenate multiple 16S amplicons? If so, what is the benefit of this? Yes, that's correct. So the benefit of concatenating these multiple 16S strands is that it takes a full advantage of hi-fi long read sequencing. So that where we can do somewhere around 18 to 25 kilobases in length. Whereas if you were to sequence just the single 16S gene fragment, that's only 1.5 kb, so you're not taking advantage of the entirety of what hi-fi is capable of. So when you concatenate multiple 16S fragments together, so in this case 12 of those, you're actually sequencing an 18 kb insert. So you're actually taking advantage of that, which makes it to where the throughput increases. And question here, I'll let Liz answer this one. We would like to customize Kinnex for ITS and V3 to V9 regions of 16S. How can we do that? Is this just adding Kinnex sequence to the barcoded primers? Like the first so many bases in the 16S primers.
Yeah, so I think if you look at the 16S protocol for Kinnex, which is on our website, and also in one of the links in the 16S app note that's part of this webinar, it shows you where you modify the universal 16S amplification with additional handles that becomes compatible with Kinnex concatenation. That is the theory. So the theory is if you have amplicons that have the Kinnex handles, you can concatenate. I want to say that based on our development experience, that theory, doesn't always translate to possibility, and there's several things to consider, such as if the Kinnex sequences have some similarities with your molecules they're amplifying, you can imagine the concatenation would fail or result in really strangely ordered arrays, secondary structures. I think the answer is yes, theoretically we have with Kinnex, we've truly demonstrated that you can do this for multiple amplicons, but our development also showed that there's a lot of optimization required to actually truly make sure that the concatenation works.
Great, thanks. On the previous thing, when I talked about cost, I just want to emphasize again that that cost that I said does not include labor, overhead, instrument, amortization, other reagents, things like that. Those are just the reagents to do, the 16S PCR plus the Kinnex kit and the sequencing reagents. So those are just inclusive of those few things. It does cost more to do other steps of the process, plus pay for labor, overhead, things like that. So if you use a service provider, you're going to get a higher cost than that, obviously. Did someone want to say anything there? Okay, I thought I heard something. Next question here regarding the microbial strain level resolution. Have people compared the strain diversity between PacBio full-length 16S and shotgun metagenomics? If yes, do you have any references? So I did share one reference when I answered the question. There are a few more cases of this too. One thing I do want to point out where it's not a publication, but we have internally compared the two data sets, shotgun metagenomics to full-length 16S, and we do see a very high correlation at the species level, at least. We didn't really look at the strain level in that case, but at the species level, we see, I think, around a 0.9 for the correlation coefficient. So they're very highly correlated between full-length 16S and shotgun metagenomics at the species level classification, which is really awesome. This is probably a good question. Maybe Ethan can answer since he works with this a lot. If not, Liz and I can probably step in as well. I'll let you take a first stab at it. We now have quite variable read counts on Sequel IIe with greater than 160 barcode pairs with our 16S workflow. Usually about 10 to 20% of the samples come out with less than 10K hi-fi reads. So this is without Kinnex. Will this Kinnex normalize the output and make future sample read counts more similar?
Yeah, right. So,
wait, sorry, is that a question for me or somebody else?
But let's let Ethan tackle it first, and then you can chime in after that.
Yeah, for sure. So for the normalization, it definitely will be very necessary to normalize the input and the amplicon product. There is depends on the sample type. You will see a certain non-specific application that you have to normalize into the product of 1.6 kb. So normalization on both ends will be very helpful with the read normalization at the read. But as with Kinnex, you actually will see much more read output. So some sample, if you have a low DNA input, you might need to either amplify a little more or get more DNA input to achieve that optimal PCR product.
I was going to ask Jocelyn, do you want to provide some input? Because we've looked, you have, of all people, have looked at quite extensively the 384 plex barcodes that we're providing in the protocol.
Yeah, so we did do 384, but with equal pooling. So with equal quantities going into your MAS, or into each of the reactions versus your indexes, we did find very even representation. I think the CV of representation was around like 7 or 8%. Don't quote me on that, but the point is it was very tight. We did not see any higher barcodes or lower barcodes. So my guess is it has more to do with how much you put into your MAS Kinnex beforehand, and if they are not equimolar, you might see more representation of the samples that are more represented before you do concatenation. I hope that helps answer and not confuse.
Yeah. Thanks both. A follow-up to that we got a question on. This is probably for Ethan in this case. Which type of DNA quantification do you use when you handle thousands of samples, hundreds of thousands? Do you use picogreen assay and plates?
Yes, so we use picogreen assay in a plate reader. So we have a module plate reader that can process thousands of samples at the same time.
Awesome, thanks. Chinu, I see that you answered this with a publication, but do you want to also voice over the question that you got about if there's any effect of probiotics in reducing necrotizing enterocolitis and preventing sepsis in preterm babies? I can't hear you. You're muted. While you work on that, I'll just go to the next. So the next question is given, and there's a similar one that just came in too on this, is, for trying to design microbiome projects where you're trying to identify both bacteria and fungi. Another question was, doing both 16S as well as eukaryotic ITS together on the same run from 2 different amplicons prepped in parallel. So basically the question is, what's the strategy for this and is this possible? So you would just basically, if you're trying to target both of those things, you'll need to use 2 different primer sets. We do have an application note that we put out a few months ago that give you some primer options for the eukaryotes or the fungal species. So I invite you to check that out on the web. You can just look it up PacBio.com and then Google like eukaryotic ITS rRNA app note. And then the 16S protocol, obviously, we just talked about today. So you would do those 2 different amplifications. You would treat them as independent pools in this case, and then you can sequence them either on the same run, but you have to be careful with the pooling based on the insert size if they are different. So it depends on like what you target for ITS and how similar it is in size, or you can run them on 2 different smart cells. So it just depends on what you want to accomplish there. Next question here for this is, I guess we can Ethan can take a stab at this if you want. If not, I can also talk on it, or Jocelyn can. For taxonomic classification, have people compared results using different databases for full-length 16S, so like GTDP, Silva, Green Genes, etc.? If yes, do you have any reference or materials?
Right. So at Zymo, I'm not sure that we have any comparison between these database, but we use our proprietary Zymo database. So we get the data from public database and we curate it to remove taxonomy and sequencing error. So is there a comparison? I still have to check if we have any reference data or paper that we can show, but at Zymo, we use a proprietary database to curate this.
Yeah, so we did some of this. So Keepin did some of this when he put together our PacBio GitHub pipeline that you can access, freely available on GitHub if you want to check it out in our Pacific Biosciences repo. Great, sorry. So what I was saying is that we actually did some of this when Keypin put together our PacBio GitHub pipeline. That's freely available online. Just check it out at Pacific Biosciences repo. But what he found is that the databases can vary widely, which a lot of you in the field know this. So what he did is he took a tiered approach. So basically utilizing one database at a time. I think it starts by default with GTDB as the best database, and then it goes to Silva to further identify those that weren't identified, and then can also go to another database if wanted, and then basically merges those results. So I don't think there's any publication that I know of. I do know that there is a group or a couple of groups working on databases specifically for this that are curating this and then trying to publish that, but that has not been published yet. I do have another RNA question that anyone's welcome to step in and answer this. That is for longer RNA. Could we concatenate only 3 genes after size selection?
So the answer is with the Kinnex kits, you are limited to the concatenation factor of 8 for Kinnex full-length RNA kit, 12 for Kinnex 16 siRNA kit, and 16 for both the Kinnex single-cell RNA and the MAS-Seq for 10x 3' kit. So the kits are configured that the concatenation factor is fixed. That said, I think not sure what the question is about how long, because if you're thinking 5 to 10 kilobases, you will still capture them based on what you've seen in Jocelyn's talk and also the Kinnex bulk app note. If you're thinking that all your transcripts are going to be over, let's say 5 kilobases long because you're doing some kind of enrichment or it's a very unusual species, then I would suggest not concatenating at all. Anything that's over an average of 5 kilobase or longer, I would recommend sequencing directly without concatenation. Revio now also supports small inserts. The Iso-seq Express 2.0 kit can be used without Kinnex concatenation. We will be providing that protocol shortly. The kit's already available for purchase.
Thanks, Liz. Gina, can you speak now?
Sure. Am I audible now?
Yes, great. Do you want to have any comment on that question that was asked for you?
Yeah, I'd like to emphasize that there are several studies which shows the effect of probiotics on not just inflammation, intestinal inflammation, but also on NEC, necrotizing enterocolitis, and sepsis. So I've given 2. One is a review article and another is again a paper. Not just that, there are a few papers which says about how these probiotic mixtures, especially single strains as well as a mixture of strains, can prevent these NEC occurrences and sepsis. Yeah.
Thanks, Jocelyn, for that great talk. Now we'll move into the live Q&A session. So I'll invite all of the webinar speakers to hop on camera and join. Again, I'll mention we're also joined by the Kinnex product manager, Liz Sang. So she is the expert. So we do have some questions that people have submitted already, but please do submit any questions that are lingering into the control panel if you have any. But I'll go ahead and get started on some of these questions now. This one was answered in the chat by, I believe, Jocelyn, but we can kind of voice over it. So could you explain why it is 40 million reads per Revio cell? Just getting confused with it being 25 million ZMWs. Jocelyn, you want to answer that and voice over?
Sure.
Or?
I thought you were going to voice over, my bad. Yeah, so the 40 million reads, and I think we talked about it a bit more in the talks after that, are segmented reads. So they're each of the segments that were connected together. So your ZMW is going to have the full concatenated hi-fi read, which will have up to 8 fragments for full-length RNA and 12 segments for 16S.
Great, thank you. So there was a question about RIN. So may I know what is the RIN required for environmental RNA samples? So maybe this is a question for, Jake, Liz, or Jeremy.
I think if it's talking about the Kinnex full-length RNA for bulk, our protocol asks that we have RIN over 7. That said, I don't actually know what the RIN numbers were in, for example, Jeremy, you had tried them in during early access. Do you remember what the RINs were?
Yeah, I mean, we typically wouldn't put anything to Iso-seq that was like less than 6, but most of ours are 7 or above probably for that.
Yeah.
But that's mostly just to prevent you from kind of just capturing a bunch of fragments, right? So.
Right.
Okay, cool, thanks. So I think this was kind of answered in the slide right after this question was asked, but we'll just ask it again just to voice it over. So this is, Jeremy, you can probably answer this. If not, Liz can chime in. Is there any size selection step that happens after the concatenation on Kinnex?
Yeah, you don't do a size selection. You just generally try to get it into the right size range with the number of concatenated fragments. I don't think there's anything preventing you necessarily from doing a size selection unless you have enough material left afterwards to load.
Yeah, we didn't recommend size selections. I don't see the need. I think mostly just removing the partial arrays would usually do the trick.
Okay, great. This one also for Jeremy. Is coverage change in the V4 and versus the Kinnex related to a bias of approaching efficiency from library to ZMW? If not, how do you think this is a difference?
I'm trying to see V4.
Go ahead. You're good.
Oh, V4. I see what V4.
Oh, I think that was referring to my slide between the different annotations, between the V4 and the V4 annotation and our current working annotation with the Kinnex for Soteria viridis. So we get yeah, there's definitely I think I mentioned in the talk that what's coming out of Kinnex is higher quality in the sense that the reads are higher quality and we definitely get more usable full-length cDNAs out. But there may be some other things that are helping out with that. There's definitely a little bit of the sort of low-end, high-end bias in which I showed on a slide in the Kinnex assemblies. And so we are getting sort of a lot more of the mid-range genes covered with more copies that helps to be able to get high quality models out from that. So I think overall the Kinnex is even in a sort of relatively good side-by-side comparison, better for us for annotation, at least for the plants and the test runs. Keep in mind this is all sort of just beta.
Great. We have other questions coming in for you now. You talked about your plan to do full-length transcriptome for polyploid. What is the ploidy level you are doing?
So a lot of the plants that we work on are tetraploids or hexaploids, and so they would have generally 2 copies, 2 subgenomes or 3 subgenomes, and then each one of those are usually most of our plants are outbred, so they'd have twice as many copies. So I hope that we can get to not only because now we're able to sort of fully resolve with PacBio Hi-Fi and Hi-C, we're able to fully resolve both haplotypes of genomes for diploid or for genomes that have 2 copies. We're able to resolve both of those. So I hope that we'll be able to maybe get there even with a lot of coverage from this from Kinnex. We'll be able to actually get annotations that are really finely tuned to not just individual subgenomes in the polyploids, but also even do haplotype specific annotations.
Awesome. Keeping it on the RNA train here, can you use Revio for, so this is probably for Liz, can you use, or Jake, can you use Revio for normal full-length RNA-Seq as the majority of library inserts should be less than 3 kb as required by Revio sequencing kit? So this is something new where they can now, right?
Yes. Yeah, so as of last week with SmartLink 13.0, customers can actually run small insert libraries on Revio as well. So I just want to say that the Iso-seq Express 2.0 kit that is upstream of the Kinnex full-length RNA kit generates cDNA, and this cDNA can actually be either concatenated and then run on Revio and Sequel 2, or you can actually directly make a SmartBell library, as you would in the past with older Iso-seq protocols. We will be releasing that protocol shortly, and with Revio now also supporting small inserts, you could, if there are specific reasons you'd like to not concatenate, you can do that as well.
Yep. The same goes for 16S as well. You can do that now on Revio without concatenating. This question can be for either Jeremy or Liz or Jake. For Kinnex full-length RNA, are there still any rRNA depletion steps performed?
I can take that first. Our official protocol does not do ribosome RNA depletion. We actually have not observed a high percentage of our RNA, especially actually because we actually bias towards getting things over 1 kb, and ribosome RNAs tend to be short. We do actually report that percentage as one of the hidden JSON files, and when I looked at them, it's generally under 20%. It doesn't seem to affect much, and it doesn't seem to warrant an extra depletion step.
Thoughts, Jeremy?
Yeah, I think that for the poly capture libraries, I think that, yeah, I mean, we wouldn't necessarily, we've never done a depletion step for Iso-seq, even when we drop it a little bit lower than that, and it's not overwhelmed by ribosome. But you know, there could be, you know, interesting other approaches. You know, doing total RNA, for example, you maybe would want to deplete some things, chloroplast, or in our case, or mitochondria genes. Could be all kinds of exciting things that you could do.
Sure. Jocelyn and or Liz, so we got a couple of questions around database and references and things, kind of similar to what we got before. Can users use custom or non-model organism transcript databases for mapping with the Kinnex workflow?
Yes, they can. We have instructions on how to get your annotation file in a way that will be recognized by our underlying tools, and that's available on our GitHub, I believe. But yes, you can, and we did it for plant genome actually.
Okay. Following up to that, are you able to add custom reference or transcripts into SmartLink version 13?
Yes, it's the same process. Yes, adding a custom reference is the annotation.
Well, you can add a custom reference genome. You can't add a custom annotation. There's a subtle difference in SmartLink where we allow custom genomes, but the only reference annotations we accept right now are the preloaded ones for GenCode for human and mouse. But if you have a custom annotation you want to use classification on, you could do so via the command line. The easiest is to go visit isoseq.how, where we have the instructions. I also want to say that as a few hours ago, we've actually uploaded the SmartLink tutorials for Kinnex, Iso-seq, and 16S on our YouTube channel. So if you just go to PacBio's YouTube channel, I'll also drop the link in the chats. We actually now have tutorials for analyzing basically the data that you're seeing tonight or your morning on YouTube.
Awesome, thank you. I think I'll shift gears over to 16S until more come in for RNA in the chat. There is something I forgot I need to disclaim. So there was some pricing mentioned, obviously, in some of the talks. I just wanted to point out that we're aware that APAC is you know, a distributor model, so the pricing may differ for various different countries. So just wanted to put that out there. So that's pricing for the Americas that we put on those. So just to keep that in mind. So this question is for Ethan. Could you please explain what might be the reason for the difference between the soil and the fecal performance? Could this be taxa abundance that explains this?
So the difference between fecal and soil basically is go all the way to the number, the amount of DNA input for each reaction, and also sorry, Liv, the DNA plate we sent to you is really mean. We have like a wide variety of condition there, but the Kinnex performed really well in my opinion. Soil sample usually have a high amount of PCR inhibitor, in which case that when you perform a target application, the amount of PCR product of the two sample type might be different, in which you will see a difference in the amount of read downstream.
Great. Following up to that is the second question here. It's about Kinnex being able to enable high multiplexing with 16S. So how does the balance of the index representation look? Is it kind of equally distributed? You did say that helps with that. Jocelyn can also speak to that after you too.
Yeah, so in our, I think I've shown some of the data on one of the slides with equimodal distribution of the different indexes. We do see post-concatenation a very good evenness of the distribution across all indexes. So we did test that in-house across multiple plates. But if you have very different inputs, I think that might make a difference on some of the representation.
You have anything you want to chime? Thank you, Jocelyn. You have anything you want to chime in on that, Ethan?
No, I think Jocelyn covers really well. As is the sample type and the amount of input really influence the normalization of read at the end. With our in-house workflow, we actually normalize the input and normalize the product before pull them and making smart bell library.
Awesome. Jocelyn, I think you answered this in the chat already, but how much species will be lost on full-length 16S amplicon sequencing? Is it less than 1 to 3%?
How much, what is lost? Barcodes?
No, just the ability to capture species.
What percentage of reads we can actually identify at the species level?
I think that's the question. It's kind of like, it asks how much species will be lost by doing full-length 16S amplicon sequencing. I'm assuming like the ability to detect species.
As compared to shotgun?
Maybe we can ask the person asking the question to clarify as compared to what?
Yeah, let me see who this was. Taka, can you clarify your question for that one, please? And then we'll come back. This question is for Ethan. Have you assessed the level of cell lysis completeness across the gram-positive or gram-negative bacteria with your DNA isolation kit?
Yes, so we do a very thorough optimization of the kit, and we have an in-house community standard with including a bacterial species range from gram-positive to gram-negative with 2 fungal species. Each species have a predetermined number of cells or the quantity in there. So we take that control, we take that standard going through our process and sequence that standard and compare with the theoretical profile. So I can share the all the information for the kit and how we validate the kit using our control.
Great. Question for Chenu. what's the evidence from out of your study that there's a need for species level resolution for the microbiome for your study?
Thanks for the question. So I would say the evidence for needing species resolution in the microbiome studies in general, it includes first is disease association, that is certain health conditions are closely related to specific microbial species. So understanding these associations at the species level, it can lead to more precise diagnostic and therapeutic approaches. And then, like I mentioned in my presentation, functional specificity, which is really important because different species within the same genus can have varying functions. And this accurate species identification, it's really important for understanding these functional rules in processes like nutrient metabolism, immune modulation, and pathogenic resistance. And another third main point is personalized medicine. So for tailoring the healthcare, knowing the exact composition of an individual's microbiome at the species level, it can improve the personalization of treatments and dietary recommendations. Hope that answers the question.
Yeah, I think so. Great. Thank you. This one's for Liz. So the number of Kinnex barcodes is currently limited to 4. Are you planning to increase that number in the future?
Currently no plans. Certainly, if you guys see a reason to increase, so we would love to hear the use case. Just so you know that we're talking about, there's 2 ways to multiplex in Kinnex kits. One is at the amplicon level and one is at the adapter level. At the amplicon level, we support 12 with Iso-seq, and then we support 384 with the 16S protocol. And then you can pull these amplicons to make a Kinnex library. Now, if you choose to mix multiple libraries together, now you have 4. The reason we chose 4 was that we believe that for cost reasons and for efficiency, most people will be doing their multiplexing at the amplicon level. So if you do, if there are use cases in which it makes sense to have more, please let us know.
There are definitely use cases where it makes sense to do that, particularly when you're comparing, different kinds of RNA isolated from different tissues, for example. If you want to maintain kind of the, well, we can talk about it later on, but yeah, because of the biases introduced in the length size, you don't necessarily want to, you don't want to barcode those on the in the cDNA level and then blend them back together again and then pull them back out again, because you'll just be introducing kind of more biases by doing that. If you want to do direct expression comparison, so that would be one reason where you might want more than 4, if you wanted more than 4 on a flow cell.
Got it.
Great. So let's circle back to that other question now that I know who actually answered in the chat. It was Chinu. So Chinu, can you voice the answer for how much species is able to not be detected or is lost with using full-length 16S sequencing?
Yeah, so about whether we lose 1 to 3% of species with full-length 16S rRNA sequencing, I would say it's not exactly straightforward, because what's cool about this method is that it's way better at catching different species than the shorter sequencing techniques. But pinning down an exact number like less than 1 to 3% isn't something the studies out there confirm. Because it depends a lot on what you're sampling. Like if it's soil, it's super complex. You might miss a bit more there. Plus the primers and the sequencing technique itself can miss some low-key species. So what I would say as an answer is in full-length 16S rRNA sequencing, the loss of species detection is generally very minimal.
Yeah, and I agree based on what I've seen come out, especially for the human gut. It's usually we're able to capture like 99-point-something% of the species in the human gut. It is vastly different for more complex sample types like soil or water or things like that, where the species diversity is very high. Where in those cases, it also really depends on the database that we're using. So if you're using a curated database that's really got a lot of those species there, then we're seeing something around like 80% or so. However, it really depends on, again, what's there and what's in the reference. So it can kind of be variable depending on the sample type and the reference databases. Did you want to, either Jeremy or Ethan, did you want to comment some on that? Because I know both of you have experience with with that, but
yeah, I think the database level is very important because with a public database, you have a lot of taxonomy error and sequencing error in there, which can result in false positive also. So comparing the data between analysis data between different databases, you might see one generate more species than the other, but that might not the true representation of the sample. Again, I want to voice Jinu's answer also is that the library prep method is very important. With a more robust library prep, you actually can enhance the application and avoid non-specific application.
awesome, thanks. There's one last question that just came in. We are getting close to time, but it's just, again, asking like we got in the earlier webinar, if there's recommendations for 16S reference databases. We've found again that it varies, like the curating the databases is always best because of like what Ethan just said, which is why, you know, Zyma does that. A lot of other folks do that themselves too, because there's a lot of noise in there and mess. GTDB we've found to be the cleanest though of all of them. However, it's going to miss out on some stuff. So where Silva might be a better approach. There's also Green Genes too. There's, you know, RDP. There's lots of different databases. So again, it's capturing all of the diversity is important. So basically meshing all those together is the best approach if you can, but it takes a lot of work on the curation side of things, obviously. And it can be quite a mess, but that's kind of a convoluted answer, but that's just how it is. No other questions that I see, so I'm going to go ahead and wrap it up here. So I'd like to thank again the speakers for their time today and for sharing their experiences and their expertise in both their talks as well as in the live Q&A here.